Every so often, a discovery is made which illuminates our understanding of ourselves and of our world.
Science advances sort of incrementally by discovering little things. But sometimes there's a big discovery that changes a lot of thinking and causes us to rethink, and Mal's part of that.
The discovery of Mal is not just central to our understanding of the immune system and how it works, but it should also help us to design vaccines that will prevent millions of people dying from infections every year.
By understanding how mutations in genes influence the immune response, we're going to be able to manipulate the immune response better. And that means we'll be able to design better vaccines and actually get rid entirely of infections like malaria and TB.
Evidence to date has suggested that this discovery of a single protein could have an enormous impact, not just on the treatment of malaria and TB, but across a range of different diseases.
So imagine we've got these three diseases which on the face of it are quite distinct: malaria, septic shock and arthritis. It turns out that our discovery of Mal is really important because it's central to all three, and this gives us the very exciting prospect to understand more about the diseases and then develop drugs to treat them.
Each of us has a unique and complex immune system, which helps us to fight disease and infection. Up to 100 years ago, almost nothing was known about it. Slowly, that has begun to change. Investigators are devoting themselves to answering the question of how exactly it works, and why some of us contract certain diseases and others don't. Luke O'Neill of Trinity College Dublin has made the study of this intricate network of defences his life's work.
Immunology is the study of how the body fights infections, mainly like viruses, bacteria, fungi. The immune system is needed to combat all those and deal with them and clear them and make us feel well again, I guess.
When a virus or bacteria threatens us, we rely on our immune system. Firstly, to recognise the invader, which may not look very different from normal cells, and then to kickstart our defensive white cells, which will come out and fight it.
So when I think of the immune system, I think of a war, because really it is an invasion by these nasty germs. These bacteria, these viruses come into our bodies and try to kill us. The immune system are the troops that come out to fight these germs. And when it's working at its best, the battle ensues, the germs are killed off, they retreat, and finally the body recovers. And this really is the best situation, the immune system doing its proper job.
However, it's the question of how our system first recognises this invader and then alerts the troops or the white cells to come out and fight, which interests Luke O'Neill.
So at the very start of the battle, we have detection of the invader, and a very special set of proteins in the immune system called the toll-like receptors are the detectors of the bacteria or the virus or whatever the invader might be. We've 10 of these in our body, and these are sufficient now to pick up that the invasion is happening, and very importantly, once they've locked onto the invader, they trigger a signal which wakes up the entire immune system, and this signal is called Mal. And it's just like an emergency flare going off, and that has the effect of stimulating the immune system, which comes out, starts fighting, and ideally that will clear the infection and the battle will be won.
The discovery of Mal in the 1990s was the culmination of many years of work on the part of Luke O'Neill and his team in Trinity College.
Our big breakthrough really was to understand this recognition process and then the turning on of the immune system. And a set of proteins were found called the tolls, or toll-like receptors. And what we found was how those tolls actually turn on a white blood cell, and that was a special protein called Mal.
The discovery of Mal offered a huge breakthrough in our understanding of how the immune system actually works. When it is working correctly, the Mal signal appears to be normal. However, for some people, their Mal signal is too strong, and it can result in chronic diseases like arthritis. Arthritis, and especially rheumatoid arthritis, is a particularly painful and debilitating disease which affects young and old alike. To date, clinicians have been forced to treat just the symptoms of the disease. But what investigators are hoping to do now is to try and target the alarm system that sets it off in the first place. In the case of arthritis, the cells which would normally kill infection are called out in huge numbers, and in the absence of any enemy, they turn on their own healthy cells and attack, resulting in the inflammation and chronic pain associated with the disease.
There's far too many troops. They're fighting themselves, they're fighting our own body, and horrible diseases like arthritis results. Now, the reason for this is the signal is now far too strong. Too many troops come out. And again, the war analogy is a good one, except in this case, it's a civil war. And now we're getting fighting against ourselves, if you like, and the body now gets inflamed. It might be joints in the case of arthritis, and the disease will then progress and get worse and worse.
One of the major centres worldwide in the fight against rheumatoid arthritis is the Kennedy Institute in London. They had a major breakthrough when they discovered one of the mechanisms that released the troops to attack their own healthy cells.
At the Institute, we are interested in why rheumatoid arthritis develops and understanding ways of treating it. And what was discovered here a few years ago was that a molecule called TNF is a major player in driving the process of rheumatoid arthritis. TNF appears to be a master switch controlling the inflammation system, and normally is produced very quickly to protect you against infection and injury. If it hangs around too long in the body, you start getting prolonged inflammation and start attacking healthy tissue like joints, as happens in rheumatoid arthritis.
A major result of the discovery of the importance of TNF was the development of the anti-TNF drug, which has been given to millions of patients worldwide. However, Brian and his team were fully aware that there must be valuable targets even earlier in the immune response.
It became clear to us that actually the original mechanism proposed couldn't be the whole answer, that it had to be more complex. And so, as with other groups, we started to ask questions of how. And what Luke's discovery of MAO really opened the field up by actually showing that it was more complex and giving that final proof. And this obviously was a major advance in the field and led to obviously further discoveries.
The anti-TNF drug was a big development in the treatment of arthritis, but it only works in about 40% of cases. The consensus now is that all investigators must look to an earlier point in the immune response. Luke is travelling to Sheffield to meet someone who has first-hand knowledge of the current drug treatments. Jerry Wilson is an investigator and clinician at Sheffield University Hospital. His patient, Linda, has an overactive MAL signal, which means that she's suffered with severe arthritis for many years. She's had many procedures and is still awaiting further surgery.
My left knee, left hip, and my toes pinned. But it's all in the pipeline, shall we say. So, part of life.
Yes. Well, as you see from in Linda's case, she's on all the conventional treatments and some of the newer expensive treatments and anti-TNF, and despite all these medications, she still has active disease and still is requiring surgery.
People ask me what it's like. Is it working? You know, I mean, Dr. Keels asked me, is it working? And my answer to him is really, I don't want to come off it to find out. I'd hate to go back to what I was like 2 years ago, where I couldn't move and I couldn't stand and I was on crutches and I just couldn't do anything.
The discovery of MAL is potentially very important for patients with rheumatoid arthritis, and hopefully the clinical use of inhibitors of MAL at an early stage in disease will be effective at preventing pain, disability, which is a feature of rheumatoid arthritis, and also lessen the requirement for surgical replacement of joints, which is frequently required.
Luke is meeting Jerry to discuss his work targeting MAL along with the toll-like receptors, which were the lookout men on the tower. This is the first point in the immune response, the earliest possible point where science can intervene to prevent disease.
Fred's as well, I guess.
There are 10 toll-like receptors, and there's no doubt that toll-like receptor 4 and MAL are the important molecules in rheumatoid arthritis. And there's a lot of evidence suggesting that inhibition of TLR4 and MAL will be very effective at treating patients with rheumatoid arthritis, particularly those who do not respond to anti-TNF. So we're very excited about the possibility of examining this pathway further.
What's the next step?
The identification of the key relationship between MAL and TOL4 has been a substantial breakthrough. The aim now is to develop drugs that will dampen down that overactive MAL signal and stop the disease in its tracks. Jerry and Luke will continue to collaborate on this pathway in an effort to fully understand the process and therefore help in the development of new treatments.
So as you can see from Linda, conventional treatments have not had a very effective therapeutic response, and therefore there is still a need for new treatments, and that's why the MAL-TLR pathway represents a way that we can treat patients who currently do not respond optimally to even the most expensive new treatments such as anti-TNF.
The field of science is fundamentally collaborative and increasingly conducted on an international stage. The normal forum for exchanging ideas and developments is on the conference circuit. Luke is giving a paper on his findings with MAL and the toll-like receptors.
That MAL story is great. That's the first time I've really seen it laid out like that. And I think the medical implications and the implications for what we are genetically are really extraordinary. Because if you have an infection that kills some people and doesn't kill others, you will have what you call a selective pressure. And that's what drives evolution, selective pressure. So what Luke and Adrian Hill are looking at, in a sense, in looking at this, is looking back at our genetic history, because they're looking at the end result.
It's often through encounters like this one between Luke O'Neill and Peter Doherty at St Andrews that research collaborations are born. So it happened when Luke met Adrian Hill. The result was a huge breakthrough in the fight against a deadly infection which linked both Luke's MAL and Adrian's work in genetics. Adrian Hill is based in Oxford and is one of the foremost researchers into the genetics of infectious diseases like malaria and TB. He works extensively in Africa, gathering samples in an effort to successfully treat this killer disease.
Malaria is the biggest killer of children today. It's killing over 1 million children a year in Africa, and we've been struggling to control that problem. We have drugs, they're somewhat effective, they only last for so long, and we have to replace them with new drugs. What would really make a difference would be in a vaccine that was effective enough and cheap enough to use widely in the developing world.
In the case of malaria, the invader arrives, but unlike what happens with arthritis, the defensive troops simply don't appear, allowing the invader to take over and kill the healthy cells. The reason the troops don't appear is because of the lack of the MAL signal.
The signal just doesn't get through. And in that situation, the infection is there, nothing is happening in the immune system, it gets a foothold, and finally disease will result. So that's one very bad scenario, the immune system just doesn't get turned on.
So you really have two ways of dealing with an infectious disease. You can treat it with drugs, or you can try and prevent it with a vaccine. So what we do with vaccines is we take a small part of the malaria parasite and generate some immunity to that, so that when the real infection with malaria comes along, the immune system is ready to tackle the infection and clear it as soon as you're exposed to malaria.
Well, we now know an awful lot more about how the immune system detects danger, how it detects viruses, bacteria, or fungi. And of course, these detection mechanisms are controlled by our genes. So you can easily imagine how a small change in any one of these specific genes could impact on how we respond to infection, how we respond to danger.
So my main interest as a geneticist over the years has been trying to find genes that affect susceptibility to malaria in African children. And we've been looking for genes that do that, but of course there are 20,000 of them, so how do you pick the ones that are most likely to be right? And when we heard about the work by Luke O'Neill on MAL, that gene looked so centrally involved in immunity that we thought it would be a great one to look at. So we then went to our DNA samples from children with malaria in Africa. We had many thousands of those, and compared the frequency of that mutation in the MAL gene in those children to the general population. And what we found was really striking. So what we're looking at here is the DNA sequence of 4 different individuals who we've studied. And you can see that the person on the top line has got an A. And if you look at the next 2 people, they've got a G, and the 4th person has got both. So 1 in 4 people have this genetic type that we've been able to show substantially protects you against getting malaria. So that's a change in the MAL gene that is central to resistance to malaria.
I guess a year later then, Adrian emailed me with some very interesting findings that really excited us. And in fact, when the email came in, I almost fell off my chair. Would have to be said, because it was such an exciting finding that he'd made. Because all my work is very much in test tubes, you know, and very much in the lab. That's my specialty. I'm a biochemist, I guess, ultimately, which breaks things down. And you always wonder, will your work ever be really relevant to real humans in real life situations? And Adrian's work definitely suggested a role for MAL in malaria.
So we were able to show this strong genetic effect of a variant of MAL. But we don't look at how those genes function. We needed Luke to do that. So he went off and came up with these very clear results. And by putting the genetic result together with all the biochemical work that Luke did, that made a really outstanding paper.
Right.
Luke and Adrian submitted their results to a scientific journal, and in February 2007, their paper was accepted and published, which was a significant achievement.
The first of many, as they say, over the moon, because it's literally years of effort, and it's so rare to get a night like this, because we spend years and years doing this, and there's many pitfalls and many sort of problems with research, and we're always held back occasionally, but to get this through is fantastically thrilling. So it's a real vindication, I suppose, and commitment by the lab, and especially for the lab, because they're the ones on the ground doing all the hard work and coming in on wet Monday mornings, you know, so it's a real reward for them as well, hence the celebration for them as well. So.
For most scientists, the only way to know that you really have something, even after the extensive tests, is when it's published in a widely recognised and prestigious scientific journal.
Every scientist wants to make a big discovery, and then once you've made what you think is a big discovery, you want to tell the world, because then other scientists can see it and they can do more work, and it extends out. And the way you do that is you publish. So you send your work off to be published in a learned journal, and depending on how big the discovery is, it's going to get published in somewhere or another. One example would be the less important discoveries possibly might go out in the local newspaper, but a really big breakthrough will appear on the cover of Time magazine. So you aim high if you think you've got a big discovery, and the highest place to aim for in our business is Nature and the Nature family of journals. They have the highest profile, they're published all over the world. Everybody will see it if it's in Nature or the Nature family journal. So that's where we went to publish our work.
Well, the Irish Society of Immunology wants to honour Irish researchers who have contributed something major to the field of immunology. And so we've instigated this award in recognition of such events. And there's major potential impact for this work. By understanding how mutations in these genes influence the immune response, we're going to be able to manipulate the immune response better. And that means we'll be able to design better vaccines and actually get rid entirely of infections like malaria and TB. This has already been shown with smallpox. Smallpox is gone from the world, but we never really understood how we achieved that. With discoveries like Adrian's and Luke's, we're going to be closer to this holy grail.
Once a paper is published internationally, one never knows what impact, if any, it might have on other investigators. One of those who read Luke and Adrian's paper was Mihai Netea in Nijmegen University in the Netherlands. He saw a possible link between MAL and his work with septic shock.
Sepsis and septic shock is the major cause of mortality in intensive care units in hospitals. The one that people will be most familiar with is MRSA. That is a bacteria that causes septic shock and kills you. So, of course, understanding sepsis and trying to come up with drugs to treat sepsis would be a tremendous thing.
Bacteria like MRSA take hold when the body is at its weakest, for example, after surgery. In a similar way to other infections, the invader is not recognised, the troops do not come out to fight, and infections like sepsis can hit our systems hard.
Sepsis is a very severe infection, actually, where the whole organism of the patient is affected. Most of the time we have to treat these patients in an ICU unit, in an intensive care unit, because of the severity of the disease. I've been treating sepsis patients for a long time, and for us it was always very difficult to understand what exactly is going on. Now, with the discovery of toll-like receptors and with the discovery of MAL, we can much better understand what exactly is going on, how can we find new drug treatments for these patients, and hopefully how can we adjust the treatment of sepsis in the future to improve the outcome of the disease.
Natea has been working further on examining the underlying links between MAL and septic shock, and he's been keeping Luke up to date with his findings.
At the moment we have had some very interesting results because we have taken the MAL forms that Professor O'Neill has discovered, and we were looking at our patients in the intensive care unit, and we have observed that one form of the MAL molecule is highly associated with a very severe form of disease and with an increased mortality, which is very significant for us because it makes us able to predict, based on MAL, which patients will have a more severe disease than the others.
Just as Hill and O'Neill found that people with a genetic variation of MAL were much more resistant to malaria, Natea can see now that patients with this variation of MAL are also less likely to develop the fatal form of sepsis. Initial results are very promising, and it offers the investigators even clearer insights into why some people die from sepsis and others don't. The target now is to design treatments which will increase the underactive MAL signal so that the immune system will call out the troops to fight the infection. Luke's original plan for visiting Natea was to discuss the MAL-TOL4 pathway in relation to sepsis. But just before he was due to leave, he had something else to show him.
Yeah, come to Nijmegen to talk to Mihai about sepsis, and then amazingly they've got this data on arthritis as well, to do with TOL4 and MAL and the whole pathway, and it tells us how important the immune system is to two different diseases. And of course, again, the hope is we could block the same pathway and not just affect sepsis, but also maybe affect arthritis. So I'm tremendously excited to hear this new data that we just heard about today.
And hopefully you can see that we have two inflamed hind paws, which are really resembles the rheumatoid arthritis patient with severe swelling. So these paws are severe inflamed, redness and swelling is clearly to see.
And you would have given the drug, as you say, maybe
three times a week.
You
can see, these animals,
you see the difference there.
The animals were treated with our novel toll receptor 4 antagonist. You can see no swelling or redness.
It's amazingly different, isn't it? Yeah, how normal they are.
Yeah.
So that's a hugely dramatic effect, isn't it? There.
Sure, there's a very good effect.
And of course, if that translates now to humans, it'll be absolutely fantastic, won't it?
I think it will be a very fantastic drug. If we see the similar effects on RA patients, that would be really a benefit for those patients.
Well, I'm blown away by what I've seen today because we've just seen this new drug in action, and it's treated these mice with arthritis, and it has a hugely dramatic effect on the mice. The first time in the world that arthritis has been blocked with a TOL4 MAL inhibitor, and it's the most exciting thing I've seen in a long time.
Natea's results have been so dramatic that already a number of drug companies are now discussing the development of treatments, and he hopes to begin human trials in 2008. Luke O'Neill's initial discovery of MAL has had an enormous impact and may even change the way we treat or prevent one of the biggest killers in the developing world.
I tell you what we need to see happen with science. What we have to achieve is a good level of global health and decent prosperity for every human being on the planet. And part of that answer, not all of it, but part of that answer has to be in medical science. And it has to be in dealing with diseases like malaria and tuberculosis.
Luke's continuing collaboration with scientists around the world has ensured that the impact of his discovery will not be confined to just one disease.
So 3 different diseases, it seems here: malaria, sepsis and arthritis, and they look quite distinct. But in fact, what unites them is the immune system. They all involve a dysfunction in the immune system. And MAL, it turns out, is a key immune protein, dysfunction in all three diseases. So of course the real excitement is there's one target for many diseases, and drugs that target MAL then and interfere with it could be used in malaria, in sepsis or in arthritis, and that would be the most tremendous thing of all.